Hello, everyone. My name is Marelyn Figueroa. I am a sophomore and my major is biology. So the topic that I will be talking about today is the effect of cyanobacteria on eutrophication of reservoirs in Brazil. The reason why I chose this topic is because in my previous short talk, I had talked about cyanobacteria on eutrophication of reservoirs in Japan. And so today I wanted to explore a different environment, which is that of northeastern Brazil. So I will be going over what cyanobacteria are, the diverse environments that they live in, how they create cyanobacteria blooms. I will also be going over the climate of northeastern Brazil. The focus of this long talk would be on the Sierra study, the methods, the data, how they compared their data to other reservoirs, the conclusions that they got on it, and how cyanobacteria blooms are managed today. So cyanobacteria are gram-negative photosynthetic bacteria that are able to fixate nitrogen. They are diverse and they're very abundant. And so 4 types of cyanobacteria that I have presented here today. The first one being, the first one being oscillatoria. And so this type of cyanobacteria is able to make trichomes, which are rows of bacterial cells that are very close together, which you can see here by the proximity. I have also included another type, which is Crococcus turgidus. And this type of cyanobacteria is able to make like a gelatin sheath around its cells. I have also included nostoc cyanobacteria. Here they have heterocysts, and these heterocysts are specialized cells that help the cyanobacteria when there is not a lot of nitrogen in its environment. I have also included anabion spiroids, which is another type of cyanobacteria that also has heterocysts. But as you can see, they have a different color from the nostoc cyanobacteria. So this shows that cyanobacteria vary, even though they could have the same type of specialized cells. Because they vary, they can also vary in the environments that they live in. For example, cyanobacteria can thrive in freshwater environments, which are lakes and rivers and ponds, but they can also live in saltwater environments like in coral reefs. They can live in cold temperatures like the Arctic, and they can also live in hot temperatures like in hot springs. So cyanobacteria are able to reproduce quickly and form cyanobacteria blooms. When they form cyanobacteria blooms, they can block off sunlight, meaning that plants are not able to produce photosynthesis and give off oxygen, meaning that animals can die because they do not have any more oxygen underwater. And so this diagram also talks about stratified water. Stratified water is water that has different layers, and the water has different layers because the layers differ in, for example, in density. The layers could differ in the amount of salt that they have, and the layers could differ in the level of temperature. And so, for example, there could be a type of stratification that can occur in lakes is thermal stratification. So in thermal stratification, the water has different temperatures. For example, it could have the top layer, and so the top layer would be the layer that is the warmest, and this is because it has more interaction with the sun, and it also has more interaction with the wind, so it has more oxygen. On the other hand, thermal stratified water can also have a bottom layer, and this bottom layer is more dense than the top layer. The bottom layer also has a lower temperature because it interacts less with the sun, and so there is less oxygen at the bottom. And so water that is thermally stratified could also have a middle layer, and this middle layer acts as a transport system to where the bottom layer could become warmer and then go at the top to the warmer water. And so this diagram is saying that stratified water increases toxic cyanobacteria blooms. And the reason behind this is that the bigger the difference between the top layer and the bottom layer, the more water the more a body of water can become stagnant or not moving. And this is because the difference is so big between the bottom layer and the top layer that the wind turbulence and the water currents are not able to mix the two layers, kind of like how water and vinegar do not mix together. So when this happens, cyanobacteria have an advantage over other organisms because they can travel from the bottom layer to the top layer. And so they can go ahead and get the nutrients from the top layer. On the other hand, other organisms that can't do that are left at the bottom layer, and they are left without oxygen, without the proper nutrients, and so they start dying off. And so this causes a body of water that becomes eutrophic. So eutrophication means that a body of water has a significant amount of oxygen that can no longer support life. So there are different factors that influence cyanobacteria blooms and that help them thrive. One of the most important factors is the excess of phosphorus and nitrogen, because this is what cyanobacteria blooms to thrive and grow. An excess of phosphorus and nitrogen, for example, from agricultural fertilizers, can come into a body of water through rainfall and through agricultural runoff. Other types of sources from from an excess of nutrients into the water include urban water runoff, residential runoff, forest runoff, and runoff from wastewater treatment plants. So all of these sources are causing different nutrients and excess of nutrients to run into the water and then eventually get into a large body of water. And in this large body of water, the organisms that were there before the cyanobacterial blooms are not able to survive because they have a decreased amount of nutrients. On the other hand, the cyanobacterial bloom uses all of the nutrients from these runoffs to expand. And so because of this, a hypoxic zone can be created. So a hypoxic zone is full of dead organisms. And so these dead organisms were no longer able to get the oxygen and nutrients they needed. So the hypoxic zone means that it is inhabitable by other organisms. And so cyanobacteria can also produce cyanotoxins. So cyanotoxins are secondary metabolites. This means that they give cyanobacteria an advantage. So it could be that in a body of water, the cyanobacteria produces the cyanotoxins. The cyanotoxins affect the organisms living nearby, they damage them. And so the cyanobacteria keep living and they keep expanding while the other organisms are damaged in the process. So there are three types of cyanotoxins. The first one, hepatotoxin. This is a toxin that damages the liver of organisms. The next one is neurotoxin. This toxin damages the nervous system of an organism. And then the dermatoxins, they are able to damage the skin and the mucous membranes of an organism. So here I've included three types of cyanotoxins. So microcystin LR falls under hepatotoxin, and it is a very known cyanotoxin because it is very strong and dangerous. Under a neurotoxin, an example of it would be anatoxin, and under dermatoxin, an example of it would be lingbiotoxin. So neurotoxins are dangerous because, again, they damage the nervous system, but they can also paralyze the respiratory muscles of an organism, causing the organism to die of respiratory failure. So a type of neurotoxin named saxitoxin was discovered when a person ate shellfish, and this shellfish was actually contaminated by cyanobacterial blooms that produce saxitoxin. So with the shellfish here being contaminated, the illness or the effect that this saxitoxin had on the person was named paralytic shellfish poisoning. And so this affects food industries because if food industries find a high amount of saxitoxin in their shellfish, they can no longer give it to the consumers because it can cause the consumers to die because of the paralyzation of their respiratory muscles. And so there has been a certain species of cyanobacteria that has been discovered to make saxitoxins. This species is named Cylindrospermopsis raceburski. And so this cyanobacteria will actually be very important in the following slides. And so northeastern Brazil is where the study took place, the Sierra study. So northeastern Brazil is characterized by having tropical semi-arid regions, as you can see in the pictures here. And so because of this, they have prolonged droughts. And so this has caused for there to be artificial reservoirs made. And these artificial reservoirs help the residents of this place. They provide water, for irrigation and for fish farming. However, previous studies done in northeastern Brazil have shown that fish farming and the building of dams have actually provided an excess of nutrients into the reservoirs, causing a high amount of cyanobacterial blooms to grow there. And so the Sierra study that we'll be focusing on is a descriptive study. It takes place in the Sierra state of Brazil over here. And so the Sierra study has suitable conditions for cyanobacteria, which include high temperatures. And there have been concerns coming from water companies in the Sierra state. And the reason is because they have found high rates of Cylindrospermopsis raceburski. And so the scientists that did a descriptive study over Sierra, they did not have, for example, experimental or control, but they were more looking into the reservoirs in Sierra state to see if their prediction was true. So they were not really manipulating any factors in these experiments, but they were more looking or observing the data that they received to confirm a prediction that they had. So their prediction was that the reservoirs in Sierra had high amounts of cyanobacteria when compared to other reservoirs. And the reason that they predicted that is because the companies of Sierra, the water companies located in the Sierra state, they had concerns about Cylindrospermopsis raceburski. They had high concerns for it, while other reservoirs located in states nearby, even though they were in the same northeastern region of Brazil, they did not have the same concerns. So their goal was to analyze the levels of cyanobacteria in the reservoirs of Sierra and compare data with reservoirs of states nearby. So the two reservoirs being studied, the first one is the Sitios Novos reservoir, and the next one is the Acarape do Mio reservoir. So the reason why these two reservoirs were chosen was because 12 months prior to the study taking place, they did tests to see if saxitoxin was present, and the test came out all positive. Also, when they checked the levels of phosphorus in these two reservoirs, they could see that they were both eutrophic because of the high levels of phosphorus. So for comparison, I have provided other states. So the Pernambuco state, this state has the Pedra Reservoir, the Botafogo Reservoir, and the Jucasino Reservoir, which will later be used for comparison. The other location is the Minas Gerais state, which will also be used for comparison of the Funal Reservoir. And so the method for this study is that they divided the time in which they would be doing the study into 3 seasons. The first is the rainy season, which is from January to June 2010. Next one is the dry season from July to December 2010. The third one is another rainy season, which is from January to June 2011. So the way they acquire their sample is by going 40 centimeters below the surface of the water and putting the water in 1 liter glass containers. Then they sedimented the organisms in a 1 liter cylinder for 24 hours. And this was to check for the concentration of cells. And then when they finally got the cells, they looked under a microscope and they stained them with 1% Lugol's iodine. So they created 3 groups to classify the organism. The first group contained Cylindrospermopsis sp. The second group contained other species of cyanobacteria. And the third group contained other components of phytoplankton. So they classified how abundant the genus that they found. They classified them into 3 groups. Dominant meaning that 50% of the density of a certain type of organism was 50% more than the average of the total density. Abundant means that the densities for this organism was above the average of the total densities of other organisms, and then frequent means that it wasn't above average, but it was less than abundant, basically. So dominant would be the organism that is more prevalent, and then under that is is abundant, and then under that is frequent. So the first thing they did is that they took data on the precipitation. So the SN reservoir right here is a bar graph showing the SN reservoir. So at the bottom is the x-axis, which is divided into the first rainy season, the middle is the dry season, and the 2011 right here, 2011/1 is the other rainy season. So then here it's comparing the 30-year average rainfall to the actual rainfall in the Sitios Novos reservoir. On the y-axis is the rainfall in millimeters. So in the first rainy season in 2013, the 30-year average, which is represented by this dark bar, was actually greater than the actual rainfall during that season. However, in the dry season, the same thing happened. There was less rainfall, but again, the 30-year average rainfall was greater than the actual rainfall. And then right here in the second rainy season, there was a difference. The actual rainfall was greater than the 30-year rainfall. So that means there was a flood in the reservoir. The same thing happened in the Acarape de Millo reservoir. So in the first season, which was the first rainy season, the 30-year average rainfall represented by the dark bar was greater than the actual rainfall represented by the gray bar. Then in the dry season, again, the dark bar, which represents 30-year rainfall, 30-year average rainfall was greater than the actual rainfall. And then in the last group, which is the actual rainfall, was actually greater, which is represented by this gray bar, than the dark bar, which means that the actual rainfall was greater than the 30-year average rainfall. So it means that there was an overflood of rainfall for both reservoirs during the second rainy season, which is in the year of 2011. So here is the data of the species found in the first group, which is Cylindrospermopsis sp. So the dominant, so the frequent species found, which is highlighted in blue, was Cylindrospermopsis ricebirski. And this was for both Sitios Novos reservoir and the Acarape de Millo reservoir. And so the most dominant organism found in the Acarape de Millo reservoir was Cylindrospermopsis raffidiopsis. And then the most abundant species found in the Sitios Novos reservoir was the Cylindrospermopsis raffidiopsis. Okay, in the group 2 are other species of cyanobacteria. Again, the most dominant species in the group 2 for Sitios Novos was Planktothrix agardi. And then highlighted in blue is the most frequent species for sitios nuevos, and highlighted in pink is the most abundant species for both sitios nuevos and acarape de Mio. And then right here on the other components of phytoplankton, the most frequent species are highlighted in blue and the most abundant species are highlighted in pink. So the most abundant species for acarape de Mio was Crucigenulia sp and Cytomonas sp, while the most frequent species for sitios novos was Alacoseria granulata. And so when they compared the percent contributions of the SN reservoir and the AM reservoir, so for the SN reservoir, on the x-axis is the time. So it represents the different, for the first rainy season, the middle would be the dry season. And over here from 2011, January 2011 to July 2011 would be the other rainy season. So here on the y-axis is the percentage of groups. So the group 2, which is highlighted by the orange color, showed a high percentage during the 3 seasons, while group 1, highlighted by blue, showed a lower percentage, and group 3 only showed a high percentage during February 11 and March, February 2011 and March 2011. Compared to that, in the AM reservoir, there was a difference. The group 1 showed a higher percentage, while group 2 showed a lower percentage, but group 3 was the same because there was a decreased amount during all 3 seasons, except in May 2011. So when they compared the concentrations of cells, again, in the SN reservoir, the group 2 was the most abundant in the concentration of cells, while group 1 was less abundant. And then in the AM reservoir, the group 1 was the most abundant and group 2 was the least abundant. And this can be seen because they, again, have the x-axis with the time and they have the y-axis with the concentration of cells. And then group 3 for both the SN reservoir and the AM reservoirs showed a lower amount in concentration of cells. And so when comparing diversity of the different types of reservoirs by the Shannon Diversity Index, on the y-axis, they had the Shannon Diversity Index, and then on the x-axis is the time in months showing the different seasons. So here you can see that the sitios novos had more diversity, even beginning in January 2010. It's around 1.0 for the sitios novos reservoir, while the Acarape do Meio reservoir has a diversity index of 0.5. So what this is showing is that there was more diversity between the 3 different groups of cyanobacteria and phytoplankton in sitios novos, while there was lower diversity in Acarape do Meio. So then when the scientists compared the results from these 2 reservoirs to other reservoirs, they found an interesting pattern, which is that sitios novos and Acarape do Meio had high concentrations of high percentages of cyanophyta, but they have low percentages of chlorophyta. On the other hand, when they compared it to a reservoir named Jucanzino, there was a lower percentage of cyanophyta by a higher percentage of chlorophyta. And so they found similar patterns of how Jucanzino had lower cyanophyta and higher chlorophyta in reservoirs named Botafogo, Pedra, and Funil. So what they found out is that cyanophyta decreased the water quality in sitios novos and Acarape de Mio, but because Jucanzino in the reservoirs of Botafogo, Pedra, and Funil had more chlorophyta and less cyanophyta, they had better water quality. And so this can also be explained by a temperature increase in the Sierra state, which actually helps cyanophyta thrive. So there had been a 2.6 degrees Celsius increase in the temperature during the last 55 years until 2017 in the Sierra state. So the scientists thought that because of this increase in temperature, there was a higher percentage of cyanophyta in Sitios Novos and Acarape do Mio. So the conclusions of the study were that the SM and the AM reservoirs did have a higher amount of cyanobacteria when compared to other reservoirs. And this is why the water companies were expressing concern for the high levels of cyanotoxins. The Shannon indices showed that there was more diversity in the SN reservoir than in the AM reservoir. And this could be due to the fact that the AM reservoir had deeper waters. So the AM reservoir was more susceptible to stable stratification, meaning that the water had become stagnant. And so this stratification favored the growth of C. raceyi in comparison to other groups. That means that group 1, which was classified by C. raceyi, actually had a higher amount than group 2 and group 3. So it had a higher amount of other types of cyanobacteria. And so destratification actually favored competition. That means that if the water was less stagnant, like in SM reservoir, then there would be higher diversity. So the rain season of 2011 showed that Cylindrospermopsis sp and had a decrease, while the group 3, which is other types of phytoplankton, increased. So what this means is that because the rainy season of 2011 caused a flood to occur in the reservoirs, the water became less stagnant in both the SN and the AM reservoir. So because there was more movement, that means that some type of cyanobacteria like Cylindrospermopsis sp could flood and escape the reservoir, while other types of phytoplankton could move up from the bottom layer of the water to the top layer and then get the nutrients that they were starving for, which means that there was an increase in group 3 phytoplankton. So the way that cyanobacterial the way that cyanobacterial blooms can be managed today is through nutrient level control. This means reducing the number of nutrient runoff that goes into these bodies of water. And this means reducing the fertilizers used in agriculture. And another way that cyanobacterial blooms could be managed is through the artificial mixing of source waters. And this is a long-term treatment in which the source waters are mixed so that the water does not become stably stratified and stagnant. And then a third way in which cyanobacterial blooms can be managed is through algicides. So with algicides, there are biocides that kill cyanobacterial cells, but by killing these cyanobacterial cells, they can cause the cyanotoxins that are inside these cells, they can be released into water. So that is why it is a short-term treatment. It is not used in rivers, but it is used in reservoirs. And the danger of using algicides is that the cyanotoxins can be released into the water sources that people use for irrigation. And so this is why algicides are used at the beginning of cyanobacterial blooms, when there are fewer cells of cyanobacteria that carry these cyanotoxins. So the key takeaways from this presentation is that the growth of cyanobacterial blooms can be increased by stratification. They can be increased by nutrient runoffs, and they can also be increased when there are more of cyanobacteria that produce cyanotoxins, such as Cylindrospermopsis sp, because these cyanotoxins give cyanobacteria an advantage over other organisms. And so they can have negative environmental effects, which means making certain bodies of water eutrophied and causing other organisms and other plants to die because of the lack of oxygen and nutrients. So there have been high concerns of cyanotoxins because they types of cyanotoxins such as saxitoxins affect humans. They affect the food industry because they can contaminate shellfish. And so one of the most important ways in which cyanobacterial blooms can be prevented, it's by controlling nutrient runoff, which could mean decreasing the amount of fertilizers that are used in agriculture. And that is the end of my presentation. Thank you.